Stingrays are usually very docile and curious, their usual reaction being to flee any disturbance, but they sometimes brush their fins past any new object they encounter. Nevertheless, certain larger species may be more aggressive and should be approached with caution, as the stingray's defensive reflex, use of its venomous stinger, may result in serious injury or death. The skin of the ray is used as an underlayer for the cord or leather wrap on Japanese swords due to its hard, rough texture that keeps the braided wrap from sliding on the handle during use. Several ethnological sections in museums, such as the British Museum, display arrowheads and spearheads made of stingray stingers, used in Micronesia and elsewhere. Henry de Montfrade stated in his books that before World War II, in the Horn of Africa, whips were made from the tails of big stingrays and these devices inflicted cruel cuts, so in Aden, the British forbade their use on women and slaves. In former Spanish colonies, a stingray is called raya latigo. Some stingray species are commonly seen in public aquarium exhibits and more recently in home aquaria. Rays are edible, and may be caught as food using fishing lines or spears. Stingray recipes can be found in many coastal areas worldwide. For example, in Malaysia and Singapore, stingray is commonly grilled over charcoal, then served with spicy sambal sauce. In Goa, and other Indian states, it is sometimes used as part of spicy curries. Generally, the most prized parts of the stingray are the wings, the cheek, the area surrounding the eyes, and the liver. The rest of the ray is considered too rubbery to have any culinary uses. Stingrays are a group of sea rays, which are cartilaginous fish related to sharks. There are about 220 known stingray species organized into 29 genera. Stingrays are common in coastal tropical and subtropical marine waters throughout the world. Some species, such as the thorntail stingray, are found in warmer temperate oceans and others, such as the deepwater stingray, are found in the deep ocean. The river stingrays and a number of whiptail stingrays are restricted to fresh water. Most myliobatoids are demersal, but some, such as the pelagic stingray and the eagle rays, are pelagic. Stingray species are progressively becoming threatened or vulnerable to extinction, particularly as the consequence of unregulated fishing. As of 2013, 45 species have been listed as vulnerable or endangered by the IUCN. The status of some other species is poorly known, leading to their being listed as data deficient. Stingrays are not usually aggressive and ordinarily attack humans only when provoked, such as when they are accidentally stepped on. Stingrays can have 1, 2 or 3 blades. Contact with the spinal blade or blades causes local trauma, pain, swelling, muscle cramps from the venom and, later, may result in infection from bacteria or fungi. The injury is very painful, but rarely life-threatening unless the stinger pierces a vital area. The blade is frequently barbed and usually breaks off in the wound. Surgery may be required to remove the fragments. Fatal stings are very rare. The death of Steve Irwin in 2006 was only the second recorded in Australian waters since 1945. The stinger penetrated his thoracic wall and pierced his heart, causing massive trauma and bleeding. The venom of the stingray has been relatively unstudied due to the mixture of venomous tissue secretion cells and mucous membrane cell products that occurs upon secretion from the spinal blade. The spine is covered with the epidermal skin layer. During secretion, the venom penetrates the epidermis and mixes with the mucus to release the venom on its victim. Typically, other venomous organisms create and store their venom in a gland. The stingray is notable in that it stores its venom within tissue cells. The toxins that have been confirmed to be within the venom are cystatins, peroxyredoxin and galectin. Galectin induces cell death in its victims and cystatins inhibit defense enzymes. In humans, these toxins lead to increased blood flow in the superficial capillaries and cell death. Despite the number of cells and toxins that are within the stingray, there is little relative energy required to produce and store the venom. The venom is produced and stored in the secretory cells of the vertebral column at the mid-distal region. These secretory cells are housed within the ventrolateral grooves of the spine. The cells of both marine and freshwater stingrays are round and contain a great amount of granule-filled cytoplasm. The stinging cells of marine stingrays are located only within these lateral grooves of the stinger. The stinging cells of freshwater stingray branch out beyond the lateral grooves to cover a larger surface area along the entire blade. Due to this large area and an increased number of proteins within the cells, the venom of freshwater stingrays has a greater toxicity than that of marine stingrays. Batoids belong to the ancient lineage of cartilaginous fishes. Fossil denticles, tooth-like scales in the skin, resembling those of today's chondrichthyans, date at least as far back as the Ordovician, with the oldest unambiguous fossils of cartilaginous fish dating from the Middle Devonian. A clade within this diverse family, the Neoselaci, emerged by the Triassic, with the best understood Neoselacian fossils dating from the Jurassic. The clade is represented today by sharks, sawfish, rays and skates. Although stingray teeth are rare on sea bottoms compared to the similar shark teeth, scuba divers searching for the latter do encounter the teeth of stingrays. Permineralized stingray teeth have been found in sedimentary deposits around the world, including fossiliferous outcrops in Morocco.